Welcome to Longevity by Design.
Hello, I'm Ashley Reaver, and I'm joined by Dr. Gil Blander. Welcome to Longevity by Design, How to Live a Longer, Healthier Life. We're produced by Inside Tracker, your science-based guide to optimizing your body from the inside out. Our guest today is Dr. Vera Gorbunova. Dr. Gorbunova is an endowed professor of biology at the University of Rochester and a co-director of the Rochester Aging Research Center. Her research is focused on the understanding of mechanisms of longevity and genome stability in studies on exceptionally long-lived mammals. Thanks so much for joining us.
You're very welcome.
So, Vera, you and I spent a few years at the Weizmann Institute of Science in Israel. I don't think that we met there, but the first time that I met you was at the Glenn Foundation presentation at Harvard Medical School a few years ago, when you present about your long-lived mammals. And I was really fascinated by the presentation and unique approach that you had. So when we started to select guest for our podcast, you have been one of the first that we said we have to have. So welcome to the show. And we'll start asking you some questions. So thank you again for joining us.
All right, Vera. So before we dive into your research, can you tell us a little bit about you? We know that you are currently living in Rochester, also working at the University of Rochester, but tell us a little bit more about yourself and the work that you're doing.
Well, my work, I guess we start from there. So it's focused on aging research. I'm using different approaches to do that. Some of them more conventional. We work with human cells, with mouse models, but we always also wanted to do something more interesting than conventional. And that's where I began looking at species that are exceptionally long-lived and using that as a research model to understand mechanisms of longevity. So we work on naked mole rat, we work on bowhead whale, which is the longest living mammal on Earth. We recently started working on bat species that are also very long-lived. And in addition, we do a lot of comparative research where we analyze, traits that are of interest using a panel of species, 20, 30, that are very diverse in their lifespans. And then we look for common trends for long-lived species. And then, the goal is to drill down to molecular mechanisms and then to see how they can be applied back to humans.
So, Vera, did you always knew that you wanted to be a scientist?
I guess yes. Somehow to me it occurred that, that was actually the only thing I knew. Obviously, there are many other jobs out there, but for some reason, nothing else seemed to be attractive enough.
I understand.
So I don't know from what age that was. My parents are physicists. So I had an example. So it's not that, it just occurred to me to be a scientist. No, I saw my parents. I knew what it is about. But I didn't want to be a physicist because that seemed to be very dry. And they were talking about some equations all the time. I couldn't imagine myself doing that. I was really fascinated by living things, plants and animals, and understanding how they work. So I wanted to do that, but this whole question, of being a scientist, I kind of thought, oh, this is like the most exciting thing. You just explore new things. What can be compared to that?
Definitely some truth there. You mentioned that your research is largely looking at long-lived mammals. Mm-hmm. So can you describe first, how do you determine what a long-lived mammal is? And what are kind of some major differences between short-lived mammals or short-lived animals in general, as well as the long-lived ones?
Well, so this is a very big question. So what is long-lived? Let's start there. there are two ways to look at it. So just simply in terms of lengths of time, you would say, well, anything that's close to human lifespan or longer, that would be long-lived. But there are not many animals that can live longer than us because we are already very long-lived. So then there is another definition which has to do also with the size of the animal or body mass. Because in general, there is this relationship that larger animals tend to live longer. So for a whale, it may be less surprising to be long-lived because generally big animals are long-lived. So then we also include in this long-lived group animals that maybe live 40 years or 50, but they're very small. So for an organism that small, living that long is exceptional. So that group would combine species that just live long, period, plus species that are very small, but they live longer than expected, I would say.
And are there differences in, I know this is what your research is looking at, but how about the aging process between long-lived and shorter-lived animals?
Well, there are many, many differences, and obviously we don't know all of them. Otherwise, we would be done with our research. I think it's easier to talk in terms of, more like life history traits, because at least this we understand a little better than molecular biology. So there are two strategies species use. So there are some species that live fast, reproduce fast, and die fast. And then there are species that take things more slowly. And the humans are clearly in the second category. It takes, like long time for young generation to mature. And then we tend to have just a few children, not hundreds of them. And then we live for a long time. So there are the species that use this strategy, but majority of the species that people use as laboratory organism actually belong to the first group. They're very fast living, very short life cycles. So I think that is kind of a key distinction. And I think to extend human aging, we really need to include those species that are more like humans into our research bestiary and study how they do it, because something that can extend lifespan of a mouse or a fruit fly may not necessarily work for the second group.
So, Vera, as we said, you use a uncommon model organism in your lab, and specifically use a lot of rodents. And if you go even more specifically, one of your favorite model organism is the naked mole rat, which live 10 times longer than mice.
That's right.
So can you elaborate a bit about that, and why do you think that it's live longer, and why have you decided to use this specific model organism?
Well, first of all, they live 10 times longer than a mouse, but they're similar in body size. They're also rodents. So that made a very nice pair for comparison. You have an animal mouse that is very well researched and very short lived. And then we have this other animal that is very long lived. So that was, of course, a very attractive factor. Naked mole rats are also resistant to almost all age-related diseases. We recently published they don't develop arthritis, even if we try to make them develop arthritis. They don't. They very rarely get cancer. They are very resistant to neurodegeneration. So this is really a model of healthy aging. And that's really what I liked about them. And then most importantly, we can keep them in the vivarium, so they're relatively easy to keep in captivity.
Okay. And you mentioned before that the longer-lived mammal are actually whales. And can you elaborate a bit about that, how long they live, and why do you think that they live so long?
Yeah, so there are many different whales, of course. The longest lived one is called the bowhead whale or Arctic whale. So they live up to 200 years. So the record
Wow.
is 211, but they could probably live longer. It's just one whale that was aged more precisely. And so they're very large whales, because whales tend to be large. Now, like, why do they live so long? So, I mean, first of all, because they are large. But those, I think, if you make like a regression line between body mass and lifespan, so expected lifespan for a whale would be around 100 years. So 200, it's still an outlier. So those bowhead whales are really, really exceptional. And why we are trying to understand. So we want to understand on molecular level. Probably on the evolutionary level, the reason they evolve such a long lifespan is that they live in fairly safe environment. Well, it was safe before humans started killing them. But if you take humans out of the picture, which was a relatively recent destruction, sort of on the evolutionary scale, those whales are very safe. They are very large. Almost no one would dare attack them. And they live in the northern waters, so there are fewer parasites there as well. So that was a very protected life for them. So they had the opportunity to evolve those molecular mechanisms to support long life. And now, like, our challenge is to understand on molecular level how they do it.
So what can we learn from those live-long animals like the whales and the naked mole rat? What can we implement in our life to live longer?
Well, I think we can learn a lot because mechanisms are very diverse, and I would classify those mechanisms. There are some that are maybe more conserved, so like we would find them again and again in long-lived animals. And then we also find mechanisms that are unique for every particular long-lived species. There is more than one. There are multiple. I can discuss some examples. Like for naked mole rat, for example, we found that they have very unique extracellular matrix. They produce very large quantities of hyaluronic acid, which you probably heard of. That is component
In makeup.
of makeup, exactly. Products.
So makeup is good.
So yeah, for naked mole rats, that works. They have very good skin. But it also
Well, you can see all of their skin since they're naked, so that's good.
Yes, so they evolved this adaptation. Interestingly, we do not find this adaptation in whale, for example. So we see this very abundant production of hyaluronic acid in subterranean species. So our hypothesis is that subterranean animals, they rub their skin on the tunnels, and they had to upregulate their hyaluronan production to get nice and stretchy skin. But then they co-opted the same mechanism for long life and like for cancer resistance, for other things. But now if you take whale, well, they didn't have to squeeze through tunnels, so they never had evolutionary pressure to upregulate hyaluronic acid, so they did something else. We are studying what that is. We have some hints that it has something to do with genome maintenance. So they may be very, very good at maintaining their DNA. And that is, again, is something like once we find how they do it, maybe we can design some interventions that can also be applied to humans to kind of reconstitute the same scenario. Because, well, we are not proposing we'll take a whale gene and make transgenic person. That would be going a bit too far. But let's say if we know which gene in the whale works differently, so then maybe we can develop some small molecules that humans could take and, kind of make them a little bit closer to the whale in that regard. So that's sort of a general idea. And with naked mole rat, that's even, I guess, easier to explain because let's say if we find we want to increase our hyaluronan production, we could develop some small molecules that slow down degradation of hyaluronan, and this way we can achieve the same effect in a human.
So going into a bit of the mechanism of aging, maybe more molecular mechanism, I know that in the past you worked on transposon, inflammation,
Mhm.
aging. So can you elaborate about that? What are the relationship between transposon, inflammation, and aging?
Yeah, so that was a very unexpected finding. I was fascinated with transposable elements, transposons, for a long time. Actually, this is what I studied at Weizmann Institute. I didn't work on aging, I worked on transposons, but in plants, so that was really far from aging. But I always kept it sort of on the back of my mind. Like, we have these genomic parasites that make up major portion of our genome, and then I decided to see what happens to them in aging. And what we found was that they kind of wake up because our cells, our body is trying to suppress them because these are parasites. So we have ways to keep them silent. But as we get older, all those protective mechanisms start to fail and transposons become reactivated. And here, our initial thinking was, well, they become reactivated, they start hopping around, and they make mutations. And this is what most people believe is the worst harm transposons can inflict on us. But it turned out, yeah, that's actually not the worst thing. The worst thing is inflammation, because transposons somehow, and we don't fully understand the mechanism, but they also manage to make DNA copies of themselves, and these copies end up in the cytoplasm. And when you have DNA in the cytoplasm, that looks as if you are infected. Like, typically, that would look like a virus invaded cytoplasm, and our cells react to that as if we are infected, and that triggers inflammation. And that may be the source of age-related inflammation that is called also sterile inflammation, because there is no obvious infection, but the infection may be coming from within.
So Vera, just for our non-PhD audience, can you explain what is transposon?
Yeah, so transposon, well, it's kind of a gene, so they were originally called jumping genes. So it's a piece of DNA that can move from one place to another in the genome. And it doesn't serve any obvious purpose for us. It's more like a parasite. That now it's not a parasite, for example, that lives in our intestines, but it's a parasite that lives in our genome. And many transposons, they originated from viruses. So it's a virus that made its way to the genome and kind of stayed there. In most cases, it lost the ability to exit the cell, so it cannot infect uh, a neighboring cell, which is what viruses do, but it can still get reactivated and within the genome. So I don't know if that was a good enough
Yeah, no, but
explanation.
that's great. It's not easy to explain. So I think that was a pretty good explanation. So my next question is about telomere and telomerase and the connection to aging. And just to give you a background, I'm in the field like 20 years, and 1 year the telomerase and the telomere are the most important reason for aging, and the next year it's just a side effect. So what do you think about it, and what is how the scientific community or the aging scientific community looking at telomere and telomerase today?
Well, yeah, so there was a lot of excitement about telomeres and telomerase probably like 15 years ago or so. And it's very elegant, right? So there are these ends of chromosomes. They tend to shrink with age, and the people were excited that may be the explanation for the aging process, that we run out of telomeres. But then there was also another side to telomeres. So people found that there is an enzyme that extends them that's called telomerase. And this telomerase is actually maybe not such a good thing because it gets reactivated in cancer cells. So most of our normal cells don't express this telomerase for some reason. Maybe there is a good reason. But in cancer cells, it gets activated. So there is this dual sort of purpose. Like, on one hand, yeah, maybe when you run out of telomeres, that may accelerate certain age-related conditions. But on the other hand, if you have too much of telomeres are too long, so then there is a risk of cancer, because in a tumor, the same scenario would play. Let's say tumor starts growing, runs out of telomeres, and it stops. So this is a very good mechanism to prevent tumors from growing. And if you lose it, so then like, oops, all of a sudden you have one less protection from cancer. So that's why it's, in the beginning, people thought, oh, well, we just extend telomeres and we live forever. But first of all, there are other things that can drive aging besides telomeres. And second, there is this danger of increasing your risk of cancer if you extend telomeres too much. So I think at this point, there are different, approaches being developed how to extend telomeres, but it's primarily limited to people that have a real deficit of telomeres. So there are genetic diseases where people are born with extremely short telomeres and they shorten much faster. Then these people suffer from like pulmonary fibrosis, for example. So there are tissues where there is very high cell turnover, they start to run out of telomeres. So for these people, that would be life-saving to develop such interventions. But for the rest of us, if our telomeres are sort of average size, I mean, it's still questionable whether we want to extend them.
It's really interesting. And you've done some research in looking at DNA repair in breast cancer cells.
Mm-hmm.
Can you kind of walk us through your research there and what you've learned from that and how it contributes to aging?
Well, so that work maybe was more focused on DNA repair and trying to find the vulnerabilities in cancer cells. So we found that there are particular pathways of DNA repair that may be enhanced in cancer cells, and we could use that to more selectively kill cancer cells. So that was the purpose of that research. As far as aging goes, so that these two are not necessarily connected. So we found one gene called RAD51 that is very important for homologous recombination, which is a type of DNA repair. It is very strongly induced in cancer cells. They probably needed to survive because cancer cells, their genomes are a mess. So they're missing all kind of checkpoints that normal cells have to maintain their DNA in good shape. So cancer cells are under constant stress and they're mutating their DNA at very high rates. So they need those mechanisms to fix themselves up, otherwise they will fall apart. So that can be explored if, let's say, we target that gene, we could more selectively kill cancer cells. So that's really what it was. I think right now cancer is still the second most common cause of death for people, and many therapies that were developed they're still very toxic. So that's kind of the tragedy of it, that people they may be cured, but many therapies would make people age much faster. So we really want to find therapies that will be not as toxic to the rest of the body and would just be killing cancer cells.
Sure. And is there anything that you feel, came from that research or other research that we as individuals could do to impact DNA repair? Maybe without pharmaceuticals. Who knows?
Well, that of course the bar is very high for interventions that you would use for healthy people, because obviously if somebody has a very serious condition like cancer, so then you would use something that is maybe toxic in a way, but the benefits outweigh the risks. But for healthy people, we should only use something that is perfectly safe, because we don't want to do any harm. And there are some strategies like that anyone can implement, for example, but that may not be very satisfying, because there are things that, well, we all already know, like eating healthy, fasting periodically. So these are sort of common interventions. We are also very interested in, when we find a gene that is worthwhile to targeting, so that there may be some natural compounds that can do it, so then people can, maybe increase their dietary intake of certain, things that are just healthy foods, but they may also be beneficial for upregulating, like DNA repair. So we published a paper last year where we gave mice, very short-term dietary restrictions. So I'm not talking about restricting calories for like months and years, a very short term, and we could see that DNA repair was enhanced. So that's again something that perhaps, maybe some people fast once a week or once in 2 weeks. That may be actually enough to give your body a little bit of a mild stress to upregulate DNA repair mechanisms.
That's very interesting. And my follow-up question is about your long-lived species. And you gave us a few examples. What are the mechanisms maybe that they adopted in order to live longer? What have you seen something like that in human, or autolog for that in human? And if not, do you think that it's possible for us to learn from them and basically extend our lifespan?
Yeah, so one conserved mechanism that we identified was DNA double strand break repair. We found that in longer lived rodents that was enhanced, and the correlation was very strong. So we could see proportionally, like the longer those species live, the better was double strand break repair. So we published it in 2019. And then we also looked at molecular mechanism, like what's different between double strand break repair between short and long-lived animals. And the main sort of player was a gene called sirtuin-6. So it's a famous sirtuin. And we found that its enzymatic activity was also higher in long-lived animals. And here we also looked at human sirtuin-6, and we saw the same thing, that it has higher activity than, for example, mouse or guinea pig. So that actually gave us confidence that SIRT6 may be a good target for looking for pharmaceutical, means of activating.
So,
And
so that's very interesting because first I done my postdoc at the Lenny Garante Lab, which found a sirtuin family. And actually last week we interviewed Brian Kennedy, which used to be the postdoc that identified the sirtuin. And he also said, like you, that SIRT6 is the one most important, most interesting as of today. So it sounds like I know a lot of scientists, and most of the time you disagree with each other. So it's really exciting to hear that two very famous and smart scientists agree with each other. So I'm very excited about that.
well, that's good to hear. Yeah, sirtuin-6, when we started working on it, well, it was like number 6. So it actually didn't get as much attention. Sirtuin-1 was, took all the fame initially. But then, when we looked at the effect on lifespan, number 6 was the most interesting because, let's say, you knock it out in the mouse, you get very severe phenotype, premature aging, genomic instability. If you overexpress, and then this is the work from Chaim Cohen's lab in Israel, so those mice live longer. So this is like your ideal longevity gene. And now we see this strong correlation across species with maximum lifespan. And we haven't published it, but this is in submission right now. We also have a version from human centenarians that shows high activity than wild type human.
Wow, that's really exciting. So you believe that somehow activation of CRT6 might be the solution?
Yes, I think that would actually help. So that may be the way to go.
And have you seen, do your long-lived mammals, like the naked mole rat and the whales, Do they have 36? And if yes, is it higher than, I don't know, the mice? Or you haven't looked at that?
Well, yes, definitely. All mammals have 36. And yes, so naked mole rat 36 is a stronger enzyme than mouse 36. So the correlation is quite striking.
It's interesting.
Yes. So actually, there are very few other processes where we would see such a strong relationship to maximum lifespan, as with double strand break repair. We also looked at other pathways of DNA repair, and we didn't find very strong correlation. But here we did. Um, yeah, I think 36 has a unique role that it both works at um, promoting double strand break repair, and it also suppresses line 1 elements that we just talked about, those transposons. So because it's also an epigenetic regulator, so it silences those parasitic sequences. Um, that's why activating it can help on both fronts, improving genome stability, also keeping parasites at bay, and in general, perhaps just keeping more youthful epigenetic structure.
Yeah, it's very interesting. I can just tell you an anecdote. When I joined Lenny's lab in 2002, we've been, I don't know, like 20 postdocs in the lab, and we had 7 proteins, 31 to 37. So most of us worked on 31 because we thought that is the most important. And then a few were maybe wiser than us, and they chose the other one. So each 2 serotonin were selected by another postdoc. So 2 and 3 for one, and 4 and 5 for another, and 6 and 7 for another. And like 10 postdoc were focused on 31, and it sounds like actually 36 is the most important, which is very interesting.
Yeah, that's a good anecdote. Well, when we started working on 36, there were maybe 1 or 2 publications about it. And right now there are so many, you cannot even keep up with the literature.
Well, if we're kind of wrapping it up here, everyone is looking for a silver bullet or something that they can do to have an impact on aging. What I've heard so far is that I should drink cosmetics, get as large as I can, and hang out in very cold water. But realistically, are there any, maybe top supplements or behaviors that you do every single day to combat aging that you could share with our listeners?
Well, I think in terms of diet, it's very good to eat fruits and vegetables, and especially wild berries. I think there is a lot of evidence that there are um, small molecule flavonoids in them that have life extending effects, and some of them may actually be activating sirtuin 6. So those, and if you eat blueberries or cranberries, that can do no harm for sure. So I think these are really important things, and that's something I do personally.
Okay, good.
Exercise is good, but in moderation, I would say. And getting good sleep, and trying to not to be too stressed. It's easier said than done, but I think one common pattern among centenarians is that they are mostly very optimistic people. Like they have very positive attitude, so there is something to that effect. Otherwise, yeah, I'm trying not to overeat. And there is also recently there was a lot of research into those feeding sort of schedules. So people are trying to restrict the time of day when they eat food, because some of the earlier works where people would see, for example, lifespan extension in mice on dietary restriction, some of this effect was explained by actually time restriction, because the mouse would consume its allotment of food and then the rest of the day it was not eating. Same for people, let's say, trying to have a longer interval, let's say, say not to eat after dark, so you eat early dinner and then don't snack until morning. So these are also very useful. I think things that anyone can implement.
Awesome. And truthfully, a lot of our guests on the podcast so far have said very similar things, but it's really nice to hear the stress management component also as something that's being repeated.
And another thing that, recently I started to realize also, in addition to fruits and berries, seaweed, interestingly linked to many health benefits. And there are components of it that, may be particularly good. So I think that, and if you look at countries with higher life average and maximum lifespan. So there are many Asian countries here, would be Japan, South Korea, and people consume a lot of seaweed. So that's another thing I would recommend.
So Vera, why do you think that seaweed is good? What is the ingredient there that make you live longer?
Well, there are ingredients in it that may actually be activating SIRT26.
So it's all coming back to SIRT6. Interesting.
Very interesting. That's a good pro tip to end on. And I'm going to go buy some seaweed probably. Vera, thank you so much for being here and sharing all of your wisdom with us. It was really great to learn from you.
Well, you are very welcome. My pleasure.
Thank you so much.
Okay, bye-bye.
And we look forward to exploring the research in the field of longevity each week with you and other leading scientists.